For thousands of years, humanity has faced enemies it could not see. Diseases crossed borders, entered cities, changed populations, and sometimes altered the course of history. Most disappeared without leaving a written diagnosis behind. But not every trace was lost. Some survived in bones, teeth, preserved tissue, and fragments of ancient DNA. Today, those remains are allowing scientists to reconstruct a hidden history of disease. Number 10. Ancient leprosy. The disease hidden inside ancient bones. Thousands of years ago, a person was buried in what is now Rajasthan, India. There was no medical record explaining what had happened to the body. No physician knew about bacteria. For millennia, the evidence remained underground. Then archaeologists uncovered the skeleton. At the ancient settlement of Balathal, researchers found unusual changes in the bones of an individual who had lived roughly 4,000 years ago. The face was especially important. There was destruction around the nasal region, and a pattern of skeletal change that specialists recognized as consistent with advanced leprosy. G.R. Robbins-Shug and colleagues investigated the remains. In 2009, they published their interpretation of the skeleton as one of the earliest known archaeological cases of the disease. That conclusion was important, but it also illustrates one of the first problems scientists encounter when studying ancient disease. Bones can show what happened to a body. They do not always reveal exactly what caused it. Several diseases can damage skeletal tissue in similar ways. A diagnosis made from bone therefore depends on the pattern, location, and combination of lesions. In this case, the changes strongly supported leprosy. But skeletal evidence and identification of the pathogen are not the same thing. Today, leprosy or Hansen's disease is known to be caused mainly by Mycobacterium leprae. The bacterium primarily affects the skin and peripheral nerves. If untreated, the loss of sensation can lead to repeated injuries and long-term physical damage. Ancient people could observe those consequences. They could not see the microorganism responsible. That changed when molecular techniques began allowing researchers to search directly for bacterial DNA in archaeological remains. Ancient DNA creates its own difficulties. After death, genetic material fragments, chemical damage accumulates. DNA from soil microorganisms enters the remains. Modern contamination can also be introduced during excavation, storage, or laboratory work. Researchers, therefore, cannot simply find a fragment that resembles Mycobacterium leprae and stop there. They need carefully controlled extraction, appropriate molecular targets, and evidence that the genetic material genuinely belongs to the ancient sample. Around the beginning of the 21st century, researchers successfully detected ancient Mycobacterium leprae DNA in archaeological human remains using methods including polymerase chain reaction, or PCR. PCR can amplify extremely small amounts of DNA until they become detectable. That opened a new path for investigating leprosy. Instead of relying only on what the disease had done to a skeleton, researchers could search for traces of the organism itself. Medieval remains from Magdalen Hill in Winchester, England, provided an especially strong example. The site was associated with a medieval leprosy hospital. Researchers examined individuals whose skeletons already showed changes consistent with the disease, but they did not rely on skeletal appearance alone. They combined osteological investigation with biomolecular methods. Mycobacterium leprae DNA was detected in several individuals, and researchers also identified chemical biomarkers associated with the bacterium. Different forms of evidence were pointing toward the same diagnosis. That is much stronger than a single clue. The next question went beyond individual patients. Scientists wanted to know how the bacterium itself had changed through time. Ancient genomes could be compared with modern Mycobacterium leprae genomes, allowing researchers to construct relationships between strains separated by centuries. The comparison revealed remarkable genetic conservation. Ancient strains were closely related to forms of the bacterium that still exist. Leprosy therefore gives scientists two histories at once. The bones reveal the effect of disease on an individual. The DNA reveals part of the evolutionary history of the pathogen. A person buried thousands of years ago can become evidence not only of illness, but of the long biological relationship between humans and microorganisms. Yet leprosy has one advantage for archaeologists. In some cases, it leaves recognizable changes in bone. The next disease often does the same, but its evolutionary history would prove far more complicated than a simple story of one pathogen following humanity unchanged through time. Number 9. Ancient tuberculosis. The infection preserved in the human skeleton. Tuberculosis can spend years inside a human body. It can damage the lungs, spread into other organs, and in some patients reach the skeleton. When it reaches the vertebrae, it can produce a destructive condition known as Pott's disease. The affected vertebral bodies can collapse, deforming the spine and leaving a pattern that may survive long after soft tissue has disappeared. For archaeologists, that pattern became one of the strongest early clues that tuberculosis existed in ancient populations. Ancient Egyptian mummies and skeletons have been especially important in the history of this research. Researchers observed spinal changes compatible with tuberculosis in remains thousands of years old. But once again, appearance alone could not close the case. A damaged spine is evidence of disease. It is not automatically evidence of Mycobacterium tuberculosis. The organism responsible was not identified until 1882, when Robert Koch demonstrated the bacterium associated with tuberculosis. That discovery changed medicine. Tuberculosis was no longer simply a wasting illness recognized by symptoms. It had a specific biological cause. More than a century later, scientists began applying molecular techniques to ancient remains. Bones, teeth, and preserved tissues could be tested for DNA belonging to members of the Mycobacterium tuberculosis complex. Researchers also used another kind of evidence. Mycobacteria possess distinctive lipid-rich cell walls containing compounds such as mycolic acids. Under favorable conditions, chemical traces of those molecules can survive even when DNA is badly degraded. The strongest ancient diagnoses, therefore, do not depend on one observation. A researcher may begin with the skeleton. The distribution of lesions suggests a chronic infection. DNA analysis searches for the pathogen. Biomarkers provide an independent molecular line of evidence. The archaeological date and burial context establish when and where the person lived. Each step reduces uncertainty. Ancient tuberculosis research has shown that forms of the Mycobacterium tuberculosis complex have affected humans for thousands of years. But its evolutionary history is not a simple straight line. Different members of the complex have infected humans and animals, and ancient genetic evidence continues to change estimates of when major lineages emerged and how they moved between populations. What remains clear is that tuberculosis has repeatedly taken advantage of the way humans live. Permanent settlements brought people into more sustained contact. Growing communities increased the number of possible hosts. Dense urban environments created additional opportunities for respiratory transmission. Human movement carried infections between regions. Civilization did not create tuberculosis from nothing, but changes in human society changed the ecological opportunities available to the pathogen. The archaeological record captures part of that relationship. A collapsed vertebra can preserve evidence of what happened inside one person's body. Molecular traces can connect that individual to a pathogen lineage. Population studies then place those infections within a much larger history of human movement and settlement. Tuberculosis demonstrates how ancient disease research works at several scales at once. One skeleton, one microorganism, one population. Then, an entire history of interaction between humans and disease. But not every major infection leaves a damaged skeleton. Some disappear from bone almost completely. To find the next one, researchers had to look somewhere else. Number 8. Ancient malaria. The disease that left its mark on human evolution. Malaria presents archaeologists with a very different problem. The parasites that cause it live primarily in the liver and blood. They do not normally produce the distinctive skeletal destruction seen in diseases such as leprosy or tuberculosis. A person could survive repeated malaria infections, or die during one, and leave behind a skeleton with no obvious sign of the parasite. So how can scientists investigate malaria thousands of years later? The answer requires several kinds of evidence. Malaria is caused by parasites of the genus Plasmodium. Among the species infecting humans, Plasmodium falciparum causes particularly severe disease while Plasmodium vivax has also circulated widely through human populations. The parasites are transmitted by infected female Anopheles mosquitoes. Once inside a human host, they pass through the liver before invading red blood cells. That cycle produces the recurrent fevers associated with malaria. Ancient written sources describe periodic fevers that historians have sometimes interpreted as malaria. The environments of ancient river valleys and warm, wet regions also created habitats suitable for mosquitoes. But neither a fever described in a text nor a mosquito-friendly landscape proves that a particular ancient person had malaria. The parasite itself was not identified until 1880. While examining blood from patients in Algeria, Alphonse Laveran observed parasites associated with malaria. 17 years later, Ronald Ross demonstrated the mosquito's role in the parasite's transmission cycle through experimental work. Modern medicine could now identify both the organism and the vector. Ancient populations left another kind of evidence, not simply evidence of the parasite, evidence of selection. In regions where malaria has placed intense pressure on human populations, certain genetic variants occur at unusually high frequencies. The most famous involves hemoglobin. People carrying one copy of a particular HBB variant, known as the sickle cell trait, receive significant protection against severe Plasmodium falciparum malaria. But inheriting two copies can cause sickle cell disease. The same variant can therefore provide an advantage in one biological context and a serious cost in another. This is evolution as compromise. Other genetic patterns tell related stories. Variants affecting G6PD are common in several populations with long histories of malaria exposure and can influence susceptibility to severe malaria. Changes involving the Duffy blood group, encoded by ACR1, can strongly reduce the ability of many Plasmodium vivax parasites to invade red blood cells. None of these genes exists because evolution planned a defense against malaria. Individuals with certain inherited traits simply had different chances of surviving and reproducing in environments where the parasite was common. Across many generations, those differences altered the frequencies of genetic variants. That means a disease can leave evidence even when it leaves no scar on bone. The record may survive in the genomes of later populations. Researchers investigating ancient malaria therefore combine several approaches. They search preserved biological material for parasite DNA. They compare ancient and modern parasite lineages. They study human genetic adaptations. They reconstruct environments that could support mosquito populations and compare all of that evidence with archaeological and historical context. The difficulty is that parasite DNA does not preserve equally well in every ancient individual. For many archaeological periods, direct molecular evidence remains much rarer than scientists would like. That makes caution essential. A region suitable for malaria is not proof of infection. A human genetic variant associated with protection is not a diagnosis of one ancient person. A historical fever is not automatically malaria. The strongest conclusions appear when independent lines of evidence converge. Malaria, therefore, adds another dimension to the history of disease. Leprosy can leave marks on the face. Tuberculosis can damage the spine. Malaria can leave part of its history in human evolution itself. Yet some epidemics leave abundant written evidence and still resist identification. One of the most famous began inside a city at war. Number 7. The Plague of Athens. The epidemic we still cannot identify with certainty. In 430 BCE, Athens was already under extraordinary pressure. The city was fighting Sparta in the Peloponnesian War. People from the surrounding countryside had moved behind the city's defensive walls. The strategy protected them from invading forces. It also created extreme crowding. Then disease appeared. Our most important witness was Thucydides. He was not observing the epidemic from centuries away. He lived through it. He became ill himself and survived. In his History of the Peloponnesian War, he recorded the progression of the disease in unusual detail. He wrote of intense fever, inflammation around the eyes and throat, coughing, stomach distress, severe thirst, diarrhea, skin changes, and profound weakness. He also observed something epidemiologically important. People who recovered appeared less likely to suffer the same disease again. Thucydides did not know about antibodies or immune memory. He simply recorded a pattern. For modern researchers, his account is invaluable. It is also frustrating. Many infectious diseases can produce combinations of fever, gastrointestinal symptoms, respiratory symptoms, and skin changes. Translating an ancient description directly into a modern diagnosis is dangerous. For more than 2,000 years, the cause of the epidemic remained a subject of debate. Then, archaeology appeared to offer a new opportunity. During excavations in the Kerameikos area of Athens in the 1990s, archaeologists found an unusual mass burial dated to the period of the epidemic. The burial practices differed from the more orderly customs normally expected in classical Athens. That made sense in the context of a sudden crisis. If these people had died during the epidemic, their bodies might preserve evidence of its cause. In 2006, Manolis Papagrigorakis and colleagues investigated teeth from individuals in the burial. Dental pulp was the target. During a systemic infection, microorganisms circulating in blood may reach the vascular tissue inside a tooth. The mineralized exterior can then protect some biological material after burial. Researchers extracted DNA and used PCR assays to test for several possible pathogens. They reported sequences consistent with Salmonella enterica serovar typhi, the bacterium associated with typhoid fever. The result attracted enormous attention. For the first time, there appeared to be direct molecular evidence, pointing toward a specific cause for the Plague of Athens. But the case did not close. Other researchers questioned whether the genetic targets used in the study could distinguish Salmonella typhi with enough specificity from related bacteria. Ancient DNA work also carries constant risks from contamination and degradation. And the clinical picture described by Thucydides does not fit modern typhoid in every detail. So, the most defensible conclusion is not that scientists solved the Plague of Athens in 2006. They found evidence supporting a Salmonella hypothesis. The precise pathogen remains disputed. That uncertainty matters. It shows the difference between a result and a conclusion. PCR can produce a sequence. Archaeology can provide an appropriate burial context. A historical account can describe symptoms. But scientists must still ask whether those lines of evidence truly identify the same event and the same organism. The Plague of Athens also shows how social conditions shape an outbreak. The disease struck when a wartime strategy had concentrated large numbers of people behind city walls. War, mobility, sanitation, population density, and biology interacted. No pathogen operates outside its environment. The epidemic affected Athens at a critical moment in the Peloponnesian War. But historians must be careful not to turn it into a single explanation for everything that followed. Disease became one pressure among military, political, and social forces already acting on the city. More importantly for this investigation, Athens teaches us that ancient DNA does not guarantee certainty. Sometimes evidence survives. Sometimes it produces a candidate. And sometimes the final identification remains beyond reach. The Roman Empire would face the same problem centuries later. Number 6. The Antonine Plague. The epidemic with no confirmed pathogen. Around 165 CE, disease began spreading through the Roman world. The empire possessed roads, ports, armies, and trade networks capable of moving people over enormous distances. Those connections supported Roman power. They also created routes along which infection could travel. One of the most important witnesses was Galen of Pergamon, one of the best-known physicians of the ancient world. His observations are the reason the epidemic is sometimes called the Plague of Galen. Galen described fever and systemic illness accompanied, in some patients, by eruptions on the skin that later formed scabs. Those details led many later scholars to suspect smallpox. It is a reasonable hypothesis. It is not a molecular diagnosis. No confirmed variola virus genome has been recovered from a person securely identified as a victim of the Antonine epidemic. That leaves researchers dependent on written descriptions, chronology, historical context, and comparisons with diseases known today. Each of those methods has limitations. Ancient medical terminology does not map perfectly onto modern diagnostic categories. The same symptom can be produced by multiple infections. Different patients can experience the same infection differently, and a historical writer may emphasize unusual or severe cases rather than describe the entire clinical spectrum. For those reasons, smallpox remains one of the leading explanations for the Antonine Plague, but the exact cause has not been demonstrated. Measles and other possibilities have also appeared in historical discussion. The responsible pathogen may remain unknown unless appropriate biological material is eventually found. What researchers can investigate more securely is the context in which the epidemic spread. Roman military campaigns connected distant regions. Soldiers returned from eastern territories. Trade moved people and goods around the Mediterranean. Dense urban centers brought large numbers of potential hosts together. Once a transmissible infection entered those networks, the empire's own connectivity could help carry it. Historical sources indicate serious disruption. The army was affected. Communities experienced loss. Economic activity came under pressure. But another caution is necessary. The Antonine Plague did not single-handedly cause the decline of Rome. The empire survived for centuries afterward. Its political and economic history involved warfare, succession crises, taxation, migration, environmental pressures, and many other forces. Disease belonged inside that history, not above it as a universal explanation. Scientifically, the Antonine epidemic is important precisely because the answer remains incomplete. We know an epidemic occurred. We have written observations from a physician who lived through it. We have plausible diagnostic hypotheses. What we do not have is a confirmed pathogen. Less than a century later, the Roman world faced another major outbreak. And this time, the surviving medical description created an even stranger puzzle. Number 5. The Plague of Cyprian, the pandemic without a known pathogen. Around the middle of the 3rd century CE, the Roman Empire entered a period of severe instability. Emperors rose and fell rapidly. Civil conflict weakened the state. External wars threatened its borders. Currency and economic systems came under pressure. Then another epidemic spread through Roman territories. Its modern name comes from Cyprian, Bishop of Carthage in North Africa, in a work usually known as De Mortalitate or On Mortality. Cyprian described a severe illness affecting the communities around him. His purpose was religious rather than medical. Yet his observations preserve some of the best surviving evidence for the outbreak. He referred to gastrointestinal distress, vomiting, weakness, problems affecting the eyes and throat, and progressive physical deterioration. Other, contemporary and later sources help establish that a serious epidemic was occurring during this period. But none tells us what organism caused it. Smallpox has been suggested. Measles has been suggested. Other viral diseases have been proposed. Some modern interpretations have even considered illnesses resembling viral hemorrhagic fevers. None has been confirmed. There is currently no pathogen genome securely linked to victims of the Plague of Cyprian. Nor is there a single archaeological cemetery from which researchers can confidently select individuals and say that these people died in that epidemic. That creates a different kind of scientific problem. With the Black Death, researchers can excavate a cemetery created during a tightly dated outbreak and test people buried there. With the Plague of Cyprian, the connection between a skeleton and the historical epidemic is much harder to establish. Even a perfectly preserved pathogen sequence would be of limited value if the archaeological context could not securely associate that individual with the event being studied. So researchers work mainly from texts, chronology, demographic arguments, and historical circumstances. Those sources establish that disease contributed to a period of extraordinary stress. They do not justify naming the pathogen with certainty. The distinction may sound unsatisfying. It is actually one of the strengths of scientific investigation. A mystery does not become solved simply because one explanation sounds plausible. The evidence has to support it. The Plague of Cyprian therefore remains an epidemic we can describe historically, but not identify biologically. And that makes the next chapter a turning point, because for another ancient pandemic, scientists finally recovered the organism itself. Number 4. The Justinianic Plague. The first plague pandemic confirmed by ancient DNA. In 541 CE, a major epidemic appeared in the eastern Mediterranean. Historical accounts describe its movement through the Byzantine world and its arrival in Constantinople, capital of the empire ruled by Justinian I. Among the most important witnesses was the historian Procopius. He described fever, severe illness, and swollen areas of the body consistent with the enlarged lymph nodes associated with bubonic plague. Other writers recorded widespread disruption. For generations, historians therefore suspected plague, but resemblance was still not proof. The breakthrough came from people buried far from Constantinople. Researchers investigating 6th century cemeteries in Europe began extracting DNA from teeth. The reasoning was the same one that later transformed many studies of historical epidemics. If a person experienced a bloodstream infection shortly before death, bacterial DNA might remain inside the dental pulp. Researchers could then recover fragments of that DNA centuries later. At sites including Aschheim in Bavaria, molecular investigation detected Yersinia pestis in individuals dating to the period of the first plague pandemic. Scientists were no longer comparing ancient symptoms with modern plague. They had genetic evidence of the bacterium. The next step was genome reconstruction. Fragments recovered from ancient teeth were sequenced and assembled, then compared with other Yersinia pestis genomes. That comparison demonstrated that the strain associated with the Justinianic pandemic belonged to a distinct branch of the plague evolutionary tree. This point becomes extremely important later. The Justinianic lineage was not simply an early version of the Black Death strain, waiting several centuries to return. The history of Yersinia pestis is branching, rather than linear. The first plague pandemic involved a lineage that appears eventually to have disappeared. The second plague pandemic, beginning with the Black Death, arose from a different part of plague diversity. Ancient genomics therefore changed the question. Researchers were no longer asking only whether the Justinianic plague was really plague. They could begin asking where its bacterial lineage belonged within thousands of years of Yersinia pestis evolution. Historical interpretation remains more difficult. Contemporary writers portray severe mortality in Constantinople and elsewhere, but the overall demographic effect of the first plague pandemic across Europe and the Mediterranean is actively debated. Some scholars argue that repeated plague waves significantly disrupted population, taxation, agriculture, and political systems. Others contend that older estimates relied too heavily on dramatic written accounts and exaggerated the pandemic's universal impact. Ancient DNA can identify a pathogen. It cannot by itself tell us how many people died across an empire. That requires different evidence. Cemeteries, settlement patterns, economic records, pollen data, historical documents, regional comparisons. A complete reconstruction depends on all of them. The Justinianic Plague therefore marks a major shift in this story. With Athens, Antonine, and Cyprian, the pathogen remains uncertain. Here, molecular evidence identifies Yersinia pestis. History and biology finally meet in the same investigation. The next disease creates almost the reverse problem. For centuries, people thought they recognized it in ancient remains. Then, genetics forced researchers to reconsider just how far back the confirmed history actually reaches. Number 3. Ancient smallpox. The disease humanity learned to erase. For generations, one Egyptian mummy stood near the center of arguments about ancient smallpox. Ramses V died more than 3,000 years ago. The preserved skin on his mummy contains lesions that have often been described as resembling smallpox. The resemblance is striking. It is not molecular confirmation. No securely authenticated variola virus genome has established that Ramses V died with smallpox. The lesions remain suggestive evidence. This distinction became much more important once scientists began recovering actual variola DNA from historical remains. In 2020, an international team studying ancient genomes from Northern Europe identified variola virus sequences in 13 individuals. 11 dated roughly from 600 to 1050 CE during the Viking Age. From 4 individuals, researchers reconstructed near-complete viral genomes. Now there was direct molecular evidence. Smallpox was circulating in Northern Europe more than 1,000 years ago. But the genomes revealed something even more surprising. The Viking Age viruses did not sit inside the same genetic diversity as the smallpox strains that survived into modern history. They formed an extinct sister lineage. The virus itself had a branching evolutionary history. Different forms of variola circulated. Some disappeared. Others eventually produced the lineages known from later centuries. Recovering those ancient genomes required far more than observing lesions. Scientists extracted fragmented DNA from archaeological remains. They screened enormous mixtures of human, environmental, and microbial genetic material. Fragments matching variola virus were identified and compared with known genomes. Where enough overlapping pieces survived, researchers could reconstruct substantial portions of the ancient virus. The result changed the evidence base for the history of smallpox. Instead of asking whether an ancient rash looked like smallpox, researchers could examine the virus directly. Humanity, however, began learning how to resist smallpox long before anyone knew what a virus was. One observation was especially important. People who survived smallpox usually did not suffer the disease again. Different societies turned that observation into a practice now called variolation. Material from a smallpox case was deliberately introduced into an uninfected person in the hope of producing a controlled infection followed by protection. The procedure could work. It was also dangerous. The recipient could develop severe smallpox. The procedure could transmit the infection to other people. Still, variolation demonstrated that humans had recognized a form of acquired protection centuries before modern immunology explained it. Then, in 1796, Edward Jenner investigated reports that previous cowpox infection appeared to protect against smallpox. He inoculated 8-year-old James Phipps with material taken from a cowpox lesion on Sarah Nelmes, a milkmaid. Phipps experienced a relatively mild illness and recovered. Jenner later exposed him to smallpox material, and the boy did not develop the disease. The experiment would violate modern ethical standards. Historically, it helped establish the principle of vaccination. Even then, eradication was still almost 2 centuries away. Smallpox continued spreading through populations around the world. Vaccination technology improved. Public health systems expanded. But the virus remained. In 1967, the World Health Organization intensified the global eradication program. The strategy depended not only on vaccination, but on surveillance. Teams had to find cases, trace contacts, identify remaining transmission chains, then create barriers of immunity around outbreaks through targeted vaccination. The final naturally occurring case was recorded in Somalia in 1977. In 1980, the World Health Assembly declared smallpox eradicated. The story is extraordinary because it runs in 2 directions. One path moves forward. Observation became variolation. Variolation helped lead to vaccination. Vaccination and surveillance eventually produced eradication. The other path moves backward. Modern genetic technology allows scientists to recover extinct variola lineages from people who lived more than 1,000 years ago. Humanity learned how to defeat the disease before it fully understood the virus's ancient evolutionary history. Smallpox could be eradicated because sustained transmission depended on human hosts. The next pathogen is different. It survives in animal reservoirs. It repeatedly entered human populations. And in the 14th century, it produced one of the best-known pandemics in history. Number 2. The Black Death. The pandemic reconstructed from the teeth of its victims. Near the Tower of London, beneath modern streets, archaeologists uncovered a cemetery that existed because of one event: East Smithfield. The burial ground was established in 1348 and 1349 as London confronted the Black Death. Hundreds of people were buried there during a tightly defined historical period. For scientists studying ancient disease, that context is extraordinarily valuable. Researchers did not have to begin by asking whether these skeletons might possibly belong to victims of some poorly dated epidemic. The cemetery itself was created during the crisis. The remaining question was biological. What was inside the victims? Medieval accounts described fever, rapid deterioration, and swollen lymph nodes. For centuries, those symptoms supported the idea that the Black Death was bubonic plague. But modern scholars still debated whether Yersinia pestis alone could explain the pandemic. East Smithfield gave researchers a direct test. The critical evidence came from teeth. In 2011, Kirsten Boss, Verena Schunemann, Hendrik Poinar, Johannes Krause, and colleagues reported the reconstruction of the Yersinia pestis genome from Black Death victims. The process began by screening archaeological material for DNA sequences characteristic of the bacterium. But identifying a matching sequence was only the beginning. Ancient DNA carries recognizable forms of molecular damage. Fragments are short. Certain chemical changes accumulate toward their ends. Researchers examine those patterns to distinguish authentic medieval DNA from modern contamination. They then use targeted molecular capture and high-throughput sequencing to recover more Yersinia pestis fragments. Piece by piece, those fragments could be aligned against reference genomes. Eventually, researchers reconstructed a medieval bacterial genome at high coverage. The argument over the pathogen changed fundamentally. Yersinia pestis was not merely a good match for the historical symptoms. Its DNA was inside people who died during the Black Death. Once researchers had the genome, another mystery emerged. What made the 14th century bacterium so devastating? Comparison with modern Yersinia pestis showed that the medieval strain was not separated from later plague bacteria by some obvious genetic feature capable of explaining the entire mortality pattern. The severity of the Black Death therefore cannot be reduced to one mutation. Host biology mattered. Environmental conditions mattered. The ecology of animal reservoirs and ectoparasites mattered. Nutrition, settlement patterns, climate, mobility, and social structure may all have influenced the scale of the disaster. The pathogen had been identified. The pandemic still required explanation. Then scientists turned toward an older mystery. Where had the Black Death lineage come from? Near Lake Issyk-Kul in present-day Kyrgyzstan, archaeological cemeteries at Karajigach and Barana contained unusually large numbers of burials from 1338 and 1339. Some gravestones referred to people dying during a pestilence. Those dates were striking. They fell only several years before the Black Death appeared farther west. In 2022, Maria Spiru, Lia Zat-Muzralina, Johannes Krause, and an international team investigated human remains from the cemeteries. They screened ancient DNA from 7 individuals. 3 people from Karajigach carried evidence of Yersinia pestis. From 2, the researchers reconstructed genomes detailed enough for evolutionary analysis. Then they placed those genomes onto the Yersinia pestis family tree. The position was remarkable. The Central Asian strain sat immediately before the enormous diversification associated with the Second Plague pandemic. It was ancestral to published 14th century Black Death genomes examined by the researchers. This did not reveal every movement of the bacterium between Kyrgyzstan and Europe. It did not reconstruct one exact caravan, army ship, or chain of animal hosts. But it moved the investigation dramatically closer to the emergence of the Black Death lineage and supported a source in central Eurasia near the Tian Shan region. The pandemic may also have left evidence in the people who survived it. Researchers compared ancient human DNA from before, during, and after the Black Death to look for changes in immune-related genetic variants. A 2022 study focusing on samples from London and Denmark identified strong signals around ERAP2, a gene involved in immune responses. Laboratory experiments suggested that one form of the gene improved the ability of immune cells to control intracellular Yersinia pestis. The findings supported the possibility that the Black Death imposed strong natural selection on human immune genetics. But evolutionary advantages can carry costs. Variants associated with stronger protection against one infection can also be associated with increased risk of inflammatory or autoimmune disease in another environment. The Black Death may therefore have left a legacy extending far beyond medieval cemeteries. The pandemic also transformed social and economic relationships, although not in one identical way everywhere. Massive population loss created labor shortages in many areas. Workers could gain bargaining power. Authorities attempted to control wages and mobility. Land use changed. But the pandemic did not mechanically produce the same outcome across every part of Europe. Existing institutions and local economic structures shaped the response. The same complexity applies to transmission. The simple image of plague moving through Europe by one rat-flea-human pathway is unlikely to explain every case. Rodent reservoirs and fleas clearly belong to plague ecology. Human ectoparasites, direct respiratory transmission in pneumonic cases, local animal populations, climate, and patterns of human movement may also have contributed under different circumstances. What remains secure is the molecular core of the story. Yersinia pestis caused the Black Death. Researchers recovered the pathogen directly from victims. They reconstructed its genome. They identified a closely ancestral strain in Central Asia shortly before the pandemic. And human ancient DNA suggests the event may even have altered the frequency of immune-related genetic variants. Yet by the time the Black Death reached London, Yersinia pestis was already ancient, much older. To find its earliest known forms, scientists had to leave medieval cemeteries and travel thousands of years deeper into the archaeological record. Number 1. Yersinia pestis, the pathogen with a prehistoric history. In 2015, researchers analyzing ancient human DNA encountered something that pushed the history of plague far beyond written records. Simon Rasmussen and colleagues studied genetic data from 101 ancient Eurasian individuals, many dating to the late Neolithic and Bronze Age. They were looking through enormous amounts of DNA, human sequences, environmental microorganisms, fragments from everything that had entered or colonized the remains after burial. Hidden among them were sequences belonging to Yersinia pestis. The team identified the bacterium in 7 individuals. Some had lived close to 5,000 years ago. Plague had been infecting humans roughly 3 millennia before the first historically documented plague pandemic. But the prehistoric bacteria were not identical to the strains responsible for later pandemics. Their genomes preserved earlier stages in plague evolution. Yersinia pestis evolved from a closely related species, Yersinia pseudotuberculosis. That transformation did not happen in one instant. Genes were gained. Genes were lost. Existing genes changed function. Different bacterial branches acquired different characteristics. One gene proved especially informative, YMT, the Yersinia murine toxin gene. In later plague strains, YMT helps Yersinia pestis survive inside the flea digestive system and contributes to efficient flea-borne transmission. Several of the earliest prehistoric plague genomes lacked it. That discovery changed assumptions about how those infections were spreading. Researchers could no longer project the classic medieval rat-flea-human model backward onto every prehistoric case. The earliest strains may have used different transmission routes. The absence of YMT does not prove that fleas played no role whatsoever, because alternative mechanisms of flea transmission can occur. But these early genomes lacked part of the genetic toolkit associated with the highly efficient flea transmission seen in later plague lineages. Another discovery pushed scientists closer to the beginning of the known plague tree. At Riņķukalns in present-day Latvia, researchers studied the remains of hunter-fisher-gatherers who lived roughly 5,000 years ago. One man, identified as RV2039, carried Yersinia pestis DNA. In 2021, researchers reconstructed the bacterial genome. It belonged to a deeply branching early lineage. It also lacked YMT. The evidence did not look like proof of a prehistoric Black Death. The researchers instead considered the infection compatible with an isolated zoonotic event, a pathogen moving from an animal reservoir into one human. That distinction matters enormously. Finding plague in one ancient person proves that the person was infected. It does not prove that an epidemic swept through the surrounding population. Scientists need population-level evidence before making population-level claims. Other prehistoric genomes revealed that Yersinia pestis was nevertheless geographically widespread across parts of Eurasia, and the bacterium continued changing. In 2018, Maria Spirou and colleagues investigated remains from the Samara region of present-day Russia dating to the Bronze Age. Researchers screened ancient teeth for Yersinia pestis using genomic mapping and metagenomic analysis. They reconstructed plague genomes from 2 individuals. This time, the genetic picture had changed. YMT was present. The strains possessed characteristics associated with a greater adaptation to flea transmission. The evolutionary transition toward later forms of plague was already underway thousands of years before the Black Death. But the story was not a ladder in which one strain simply replaced another. Different plague lineages existed. Branches separated. Some vanished. Others survived long enough to contribute to later diversity. This is why the Justinianic Plague cannot be treated as the direct ancestor of the Black Death. Both pandemics were caused by Yersinia pestis. Their pandemic lineages belong to different branches. The lineage associated with the first plague pandemic appears to have died out. The second plague pandemic emerged centuries later from another branch of bacterial diversity. The pathogen survived because plague does not exist only inside humans. Yersinia pestis circulates within ecological systems involving wild mammals and their parasites. A lineage can persist within animal reservoirs even when large human epidemics disappear. Then, changing ecological or social circumstances can create another opportunity for transmission into people. Humans did not see the bacterium itself until the 19th century. During an outbreak in Hong Kong in 1894, Shibasaburo Kitasato and Alexandre Yersin independently investigated the organism associated with plague. Yersin produced a particularly accurate description of the bacillus recovered from affected humans and rats. The organism would eventually be named Yersinia pestis in his honor. But discovering the bacterium still left another question. How was it moving between animals and humans? Researchers had repeatedly observed that rat die-offs were associated with human plague outbreaks. Attention turned toward fleas. By the late 19th century, researchers including Masanori Ogata and Paul-Louis Simond were investigating flea transmission. Simond's experiments in India in 1898 became especially influential in establishing the rat-flea hypothesis, although later experimental work was needed to clarify and strengthen the mechanism. Modern microbiology eventually explained how plague can circulate among mammal hosts and fleas. Ancient genomics then revealed that this transmission system itself had an evolutionary history. The bacterium inside a 5,000-year-old hunter-gatherer was genetically different from plague bacteria circulating during the Black Death. Some early strains lacked adaptations present in later ones. Bronze Age lineages acquired new biological capabilities. Pandemic branches appeared and disappeared. That is one reason plague could never be eradicated in the same way as smallpox. Variola virus depended on humans for sustained natural transmission. Break every human transmission chain, and the virus has nowhere else to circulate. Yersinia pestis can persist in animal reservoirs. Human cases can disappear while the bacterium remains in nature. The story of plague is therefore not simply the history of one bacterium repeatedly attacking humanity. It is an ecological history. Humans, animals, vectors, climate, movement, settlement, and a microorganism changing within that system for thousands of years. Ancient DNA allows scientists to see parts of that process that no historical writer could have recorded. A tooth from a prehistoric grave can preserve fragments of bacterial DNA. Those fragments can be extracted in laboratories designed to reduce contamination. Their molecular damage can be examined to test whether they are truly ancient. Researchers can sequence them, reconstruct genomes, compare mutations, build phylogenetic trees, then place one infection from 5,000 years ago inside a biological history extending into the present. But even that technology does not reveal everything. Ancient DNA preserves unevenly. Cold and dry environments may retain genetic material far better than hot or humid ones. Only a tiny fraction of ancient people have been excavated. An even smaller fraction have undergone molecular testing. And failing to find a pathogen in one skeleton does not prove that person was never infected. The ancient disease record is a sample, not a complete archive. Every new genome, therefore, has the potential to change the story. A newly excavated individual may push a lineage farther back in time. A previously unknown strain may appear on another branch of the evolutionary tree. A genetic feature thought to have emerged at one moment may turn up centuries earlier. And a historical theory that looked convincing may fail when biological evidence finally becomes available. That is what makes ancient disease research so powerful and so difficult. Across these 10 investigations, the evidence changed form again and again. Leprosy left patterns in bone and bacterial DNA. Tuberculosis left skeletal damage and molecular traces. Malaria revealed how disease pressure can become visible in human genetics. Athens produced a possible molecular clue without a universally accepted diagnosis. The Antonine Plague and the Plague of Cyprian remain historically documented epidemics without confirmed pathogens. The Justinianic Plague crossed the line from historical suspicion to genetic identification. Smallpox showed that ancient genomes can recover viral branches that have completely disappeared. The Black Death turned a medieval cemetery into a molecular record of one of history's greatest pandemics. And prehistoric Yersinia pestis revealed that the plague story began thousands of years before any writer could record it. Together, they reveal a larger pattern. Disease was never separate from civilization. When humans built permanent settlements, the ecological conditions of infection changed. When populations became denser, some pathogens gained more opportunities for transmission. When merchants, armies, and migrants traveled farther, microorganisms traveled with them. When environments changed, vectors and animal reservoirs changed too. And when disease placed enough pressure on human populations for enough generations, human genetics could change in response. The history of civilization is therefore also a biological history, a history written partly in buildings and documents, and partly inside the bodies of the people who lived through it. Those people never knew that their teeth might preserve bacterial genomes. They never knew that changes in their bones could become diagnostic evidence thousands of years later. They never knew that scientists would reconstruct microorganisms their own societies could not even see. Modern science can now read some of that hidden record. Not perfectly, not completely, but with every ancient genome, every archaeological discovery, and every carefully tested hypothesis, the invisible history becomes clearer. Ancient pathogens tell us where disease came from. They reveal how human societies created opportunities for transmission. They show how microorganisms evolved alongside changing populations and environments. And they remind us of something that has been true since long before recorded history. Humans have always been changing the world around microbes, and microbes have always been changing us.